So the next thing we need to look at is leukocytes. Leukocytes. Another word for leukocyte is the white blood cells. White blood cells. We said that red blood cells are the non-nucleated formed elements of the blood, which means red blood cells have no nucleus. But white blood cells are the nucleated formed elements of the blood, which means they have nucleus. Sometimes the nucleus are multi-lobed, bilobed, or they could be single, depending on the type of white blood cell. So white blood cells refers, are we writing? White blood cells refers to the colorless and nucleated formed elements of the blood. They are larger in size than the red blood cells, but they are lesser in number than red blood cells. So we have more red blood cells in the human body than the white blood cells. Alright? Types of classes. There are two major classes of the white blood cells. Number A are known as granulocytes. Granulocytes. It's on the slide, so you don't ask me to spell. Number A are the granulocytes. And then number B are the agranulocytes. So what are granulocytes? And what are the agranulocytes? Granulocytes refers to white blood cells with fine or coarse granules in their cytoplasm. Did you get that? I said granulocytes are white blood cells with fine, F-I-N-E, fine, beautiful, fine. Another word for fine in this context is small, alright? So when you hear fine, it means they have small granules in their cytoplasm. I'm going to show you some pictures. So when I said granulocytes are white blood cells with fine, in parentheses, small or coarse. Coarse simply means large, so you can add that in parentheses. Granules in their cytoplasm. However, the agranulocytes are white blood cells that are completely devoid or lacking either fine or coarse granules in their cytoplasm. So the cytoplasm of the agranulocytes are clear. They lack small or large granules in their cytoplasm. Going forward, let's look at granulocytes in details. Alright? I said granulocytes are white blood cells with small or large granules in the cytoplasm. Now, there are three major types of granulocytes. There are three major types of granulocytes. The first one is known as the neutrophils. Number B are the eosinophils. And then number C, we have basophils. You are conversant with this, right? I hope you are conversant with this. You've not heard of neutrophils, basophils, eosinophils, lymphocytes, and monocytes before? Did you not do biology in secondary school? Okay, you didn't go to secondary school. I'm not surprised. Because some of you even came through special centers. We don't have special centers in university. So if you like, they play. Okay. We'll look at the three major types of granulocytes, beginning with neutrophils. The first diagram on the slide shows you what neutrophils look like. Neutrophils are white blood cells with fine granules in their cytoplasm. So these neutrophils contains tiny or small granules in their cytoplasm. And these granules can either stain with red and blue dye. I hope you get what I mean. Examine the structure very well. Okay, you have the cell here. And you have, this is the cytoplasm. And then you can see something like dots in the cytoplasm. These dots. Now, some of these granules or tiny dots are red in color, right? While some are blue in color. Alright? Now that tells you that these granules can stain with both red dye and blue dye. And this is where I've been emphasizing on practical classes, because these are some of the things you should do in the lab. Because when you get there, under blood physiology, you can get white blood cells, but you have to learn how to collect blood. First thing, collection of blood. You do it yourselves in the lab. Don't you want to become doctors? It starts from physiology lab. Because we are going to give you syringes, we'll give you the tourniquet, and then we'll group you. In that group, one or two persons will become the patients, and then The rest will become doctors. Alright? Good. Yes. And then the patient will, bring his or her hand. And then the doctors in the group will use the tourniquets to, tie around the hand. And then use the syringe to collect blood from the patient's vein. Okay? And when you have done that, the microscope will be there. For each room, you have your microscope. And then you put that blood sample on the slide and you view. You should be able to identify the blood cells, red blood cells, white blood cells, the different types of white blood cells. And when you examine neutrophils under a microscope, you would notice the major things you would notice are two. First, the nucleus. Look, you will notice that neutrophils have multi-lobed nucleus. Alright? If you count the nucleus on the slide, you will see that this is one. Okay, let me use red. You will notice that this cell has 1, 2, 3, 4 nucleus. So that is the first thing you will notice. That the neutrophils, they have multi-lobed nucleus. So they have 3 or more nucleus. First observation. Secondly, you look at the cytoplasm, and then you will notice that the granules are tiny, and then they stay with red and blue dyes. It's not rocket science. So when we tell you, get your manuals, do practicals, you think we are stressing you so much. You must be able to identify all of these things. It starts from now. Okay. So that's for neutrophils. Because you are going to see other types of cells in the blood that you have collected from your patients in the lab. But the onus is on you to be able to differentiate the types of blood cells. You need to know whether this is neutrophil, or whether it is basophil, or whether it's lymphocytes, or whether it's monocytes. If you cannot do this thing now, it means that you will not graduate as a professional doctor, but you will graduate as a native doctor. Yes. So instead of you to go to the hospital, we'll send you to your village, where you'll be missing halves for the village people. Alright, that is for neutrophils. The second one is the eosinophils. I have said neutrophils have fine granules in the cytoplasm, which stains with what? Red and blue dye. And they have multi-lobed nucleus. Later on, we'll come to their functions, and then we'll look at the pathologies. But going forward, eosinophils. These eosinophils are white blood cells with coax or large granules in their cytoplasm. I said eosinophils are white blood cells with large granules in the cytoplasm. And these granules stains with red dye. Alright? So if you observe the cells properly, you will note that the granules are big and they stain with only red dyes. Good. Eosinophils have bilobed nucleus. That is, they have two nucleus in their cytoplasm. Is that clear? The basophils are white blood cells with large granules in their cytoplasm which stains with blue dye. Alright? So these cells stains with blue dye. Yes, the granules are large, just like that of the eosinophils, but the difference is that granules of the eosinophils stains with red dye, while that of basophil stains with blue dye. These basophils also have bilobed nucleus. But the difference here is that their nucleus is kidney shaped, alright, or horseshoe shaped. Look, you have this for eosinophils. Then this is one nucleus, this is the second nucleus. And then you have this for basophils. If you examine the nucleus of basophils, you see that they are kidney shaped. Have you seen a beans? Beans, the normal beans you eat. If you pick beans, you see the shape. So it has the shape of the human kidney. Have you seen the structure of your kidneys? Have you seen the structure of your kidney? Your own kidney. That is why practical classes are important. Because in anatomy practical, one of you will lie down on the lab bench and will dissect your tongue. We are going to dissect your tongue. One of you will lie down. I'm serious. It's practical sections. In anatomy practical, when you are going for dissection, one of you will donate yourself. You lie down and then we use dissecting sets to open your tummy, open your belly, and we begin to identify the various organs, know their shape, know the cells, and all the blood vessels supplying them. And when we finish, we close the stomach. You will not die, don't worry. Are you scared of dying? No. That is medicine and surgery for you. So you need to prepare your mind. So going forward, the agranulocytes are of two major types. We said that agranulocytes are white blood cells that lack granules in the cytoplasm. Alright? So there are two types of the agranulocytes. The first one is the monocytes. Alright? Number one, monocytes. And then the other type are the lymphocytes. Lymphocytes. So we begin with the first one. Monocytes are white blood cells with large nucleus in the cytoplasm They are white blood cells with large nucleus in the cytoplasm, which are kidney shaped. Okay? The cytoplasm of monocytes does not contain fine or coarse granules, so it is clear. Is that taken? And then the other one is the lymphocytes. Lymphocytes are also cells that lacks fine or coarse granules in the cytoplasm. The nucleus are also large and they occupy all of the cytoplasm. There are two major types of lymphocytes. The first one is known as T lymphocytes, and the second one is known as B lymphocytes. I said there are two main types of lymphocytes. Number A, T lymphocytes. Number B, B lymphocytes. The T lymphocytes were first discovered in the thymus gland. I said T lymphocytes were first discovered and they were thymus gland. Alright? Hence the name T lymphocytes. So that is where they derive their names from. Thymus gland. Alright? However, the B lymphocytes were first discovered in the bursa of Fabricus in birds. Alright? The bursa it is spelled B-U-R-S-A F Fabricus. You know how to spell it now, so you don't stress it. Bursa of Fabricus. You have F-A-B-R-I. This is I. Then C-U-S. Bursa of Fabricus in birds. Birds. You understand what I mean by birds? I don't know how to pronounce birds. Not the one you lie down at home, the ones flying in the air. Yes. So these B lymphocytes were first discovered in the bursa of Fabricus in birds. Hence, the name B lymphocyte. Okay? Alright, are we tired? We still have a lot of time. You don't have another class today, right? Which practical? Huh? Which one? Which of the practicals? Physiology? Histology? Okay, let's look at the sizes, lifespan, sizes and lifespans of white blood cells. At least before we go, we can quickly run through this. So we begin with the neutrophils. Neutrophils have a diameter of 10 to 12 microns. Alright, the diameter. So they have a diameter of 10 to 12 micron, and their lifespan is between 2 to 5 days. So whenever they are mobilized into the circulation, they can only exist for 2 to 5 days, minimum of 2 days and maximum of 5 days. Okay. For the eosinophils, their diameter is 10 to 14 microns, and their lifespan is between 7 to 12 days. For that of the basophil, they have a diameter of 8 to 10 microns, and a lifespan of 12 to 15 days. Monocytes happens to be the cells with the largest size. So they have a diameter of 14 to 8 microns and a lifespan of 2 to 5 days. For lymphocytes, the diameter is 7 to 12 microns. And they can only live for about half a day, minimum of a half day, and maximum of 1 day. Properties of white blood cell. Very quickly, properties of white blood cells. White blood cells have the following properties. The first one, diapedesis. What is diapedesis? Yes? What is diapedesis? It means they can squeeze through tiny vessels. Okay, that is diapedesis. Now you need to go and look at the properties of white blood cells. Okay, but I'll just mention them for you: diapedesis, amoeboid movements, chemotaxis, and phagocytosis. Okay, go and look at the meaning of all of that. Okay, functions of white blood cell. When is that practical? 12 o'clock. Right? Okay, functions of white blood cells. Functions of white blood cells. Generally, white blood cells play significant role in the defense mechanism of the body. So they help to fight and destroy invading pathogens. Okay, everything that has to do with defending the body against bacteria and pathogens fall under, in fact, are the roles of white blood cells. So the neutrophils plays an important role in defense mechanism of the body. How? They play an important role in defense mechanism of the body. Actually, they try to engulf invading bacteria and pathogens. Alright? So they, as soon as they see any foreign substance in the body, they are mobilized and they try to engulf those substances and also try to disintegrate them or make them inactive. The Eosinophils plays an important role in defense mechanism of the body against parasites. These ones attack parasites. Alright? Especially those ones that are too large to be engulfed by the neutrophils. So they will attack them and disintegrate them or make them inactive. For the basophils play an important role in healing processes. So their number increases during healing processes. Alright? Basically, their functions is in hypersensitivity reactions. So the basophils plays an important role in hypersensitivity reactions by secreting some neurotransmitters. Alright? You will get to know that soon. Monocytes are larger cells among the leucocytes. So just like the neutrophils, the monocytes are also very motile, that is, they move. And they play phagocytic roles. Phagocytosis simply means cell what? Cell what? Eating. So what they do is that as soon as they see an invading bacteria, they move towards them and they swallow them up. Alright? These cells wander freely through all tissues of the body. That is for the monocytes. The lymphocytes plays an important role in immunity. Immunophysiology is something you will do also in this class. That's immunophysiology, when you begin to talk of passive and active immunity. Alright? You begin to learn about the roles of cytokines and all other things, all other cells that plays important role in immunity of the body. Okay, can you see the slide? Okay, this slide shows you a table, and the table is about pathologies of white blood cells. Alright? Pathologies. The first one we have here is neutrophilia. Neutrophilia. Generally, when you have leukophilia or leukopenia, they mean two different things. Leukophilia simply means increase in white blood cell count. Leukophilia, whereas leukopenia simply means decrease in white blood cell count. So this will guide you. Remember, we have different types of white blood cells. So neutrophilia refers to increase in neutrophil count. Alright? And it can occur during acute infection, metabolic disorders, injection of foreign proteins, even injection of vaccines. Alright? The neutrophil count increases in such situations. However, in neutropenia refers to decrease in neutrophil count. And this can be seen in bone marrow disorders, tuberculosis, typhoid, and autoimmune diseases. Okay? So the next one is eosinophilia. What does it mean? What? Increase in eosinophil count. Okay? And that can occur in allergic conditions such as asthma. Alright? In asthmatic patients, if you take their blood samples, you find out that there is increase in eosinophil count. Okay? And there are other things that can increase eosinophil counts. Alright? Like blood parasitism. Okay, and then we have eosinopenia, which refers to decrease in eosinophil count. Just like that, basophilia is what? Basopenia is what? Okay. Alright, so you go and look at conditions that can cause increase in white blood cell count and those conditions that can lead to decrease in white blood cell count. Good. Our next topic for today is the physiology of thrombocytes. Physiology of thrombocytes. Another word for thrombocytes are the platelets. Are you tired?
Yes.
Are you hungry?
Yes.
Okay, should we take a break?
Yes.
Where? Where is the venue? ShopRite. ShopRite? Okay. We'll stop here for today. So in our next class, we are going to look at physiology of thrombocytes. I think we are bringing on far.